Hello, welcome to MACSMIN again. Let me now introduce William Jeffcott from our group. He will talk about continuous invariant-based maps for the protein data bank. Please, Will.
Okay. Thank you for the introduction, Ali. Sorry, there you go. So yeah, my talk today will basically be about continuous invariant-based maps of the protein data bank and builds on what Olga discussed in the last talk, really. So there'll be some sorts of reference to that. So yeah, as I say, title, continuous invariant-based maps of the protein data bank. And the abstract for this talk is, in the past, basically the PDB was largely visualized by using 2 or 3 moments of inertia of proteins. At the level of amino acids, it was usually visualized by using Ramachandran plots of the 2 torsion angles. And in this talk, we present continuous maps of the latest PDB version at the level of chains and residues using complete invariants consisting of 9 invariants per residue, or looking at the protein backbone, which is the sort of 3 main atoms per residue of carbon, alpha carbon, and nitrogen. The resulting maps basically show quite large variations in the geometric invariants. So, for example, bond lengths and bond angles. And then we also present some examples of geometric chameleons. So they're the proteins with the same primary structure, but differing tertiary structures or geometries. So begin by introducing the LAI invariants, and we already touched on this in the last one, so I'll skip over this relatively quickly. I think the diagram that I have has the correct ones this time, but again, that was just a lucky dip based on getting off Google. Yeah, but there's basically, for each residue in the backbone chain, there are 9 LAI values. So in total, the LAI matrix has dimensions m by 9. And then these are the 3 bond lengths, so length N, length A, and length C, as we refer to them here. So those are the differences between N and A within the same residue, A and C within the same residue, and then C and then N in the next residue, respectively. Then we've got angle N, angle A, and angle C. So these are the angles that sort of form at the named atom. So angle N, for example, is the carbon from the previous residue, the nitrogen from the current residue, and then the alpha carbon from the next residue, from the current residue as well. And this is the angle formed. So we take that angle as well in the interval 0 to 180. So we take the shorter angle. And then lastly, there's the 3 torsion angles. So we've called them here tau NA, tau AC, and tau CN. These are basically the angles formed between the 2 intersecting half planes, which span triples of atoms. So yeah, in the case of tau NA, we've got the triple CI minus 1, NI, AI. So that's one of the half planes, and then the other half plane is NI, AI, CI. And we measure the angle between these. So these are conventionally named phi, psi, and omega in literature. And the sort of equivalences here. So our tau NA is what's normally phi, tau AC is what's normally psi, tau CN is normally omega. And generally in the literature, these angles are stated in the interval minus 180 to 180 using the right-hand screw convention along the chain. So some of our definitions are going to differ slightly, but I'll go into that as we go along. Wait, is it? Okay. I thought it was different to. I thought this one was different to Olga's. It is. But it's wrong again. Ah, okay. Maybe I'm not right then.
Yeah.
So yeah, we're going to phase about some of the distribution plots of the LAI invariant. So we basically, as Olga referred to in a previous talk, we clean the entire protein data bank to get sort of rid of erroneous cases and to take the sort of 20 more standard amino acid cases. And this left us with 704 K different chains, which roughly corresponds to plotting the geometry of 109 million residues, which we can see the counts of here on the right. So we've got ordered from sort of least common to most common, the residue, and then the respective counts and the percentage of the total data frame that we looked at. And across the next slides, we'll basically see the variation within each of these invariants and see that the certain geometric configurations are more common than others, but in order to fully capture the geometry of the protein, we do need to consider all 9. So if we begin by looking at length distributions, so here we've plotted histograms for the 3 lengths. So on the y-axis, we've simply got the number of residues. On the x-axis, it is length n, length a, and length c. And then there's some summary statistics below, so we can see the means for each of the 3 lengths in angstroms. See the standard deviation of the peak, which is sort of relatively low in all 3 cases. Then we've quoted a percentage of cases that lie within 3 standard deviations of the mean. And then also we've quoted a percentage that lie within plus or minus 0.01 angstroms of the mean. So we can see, yeah, from these histograms, basically the variation in each of the lengths. So it's worth noting that because we've got a log scale, the sort of tails on the left and right look comparatively big here. And that in general, for these distributions, the standard deviation is very low. But we still see plenty of sort of outlier cases. So there's about 98% within three standard deviations of the mean, but there's still sort of, well, 1.8% in this example outside that. And that constitutes, because we have 110 million in total, that constitutes, you know, in the order of millions of residues that have sort of non-standard lengths in this case. And then we can go on to plot these heat map charts. So for the previous histogram plots, we might ask the question of, is there any correlation between the various bond lengths? So, for example, if we had a uncharacteristically high length n in one plot, are we seeing that in the other lengths? Well, plotted the same one. I know, sorry. Yeah, so we basically do a 2-way plot of the lengths. So here for the first one, we've got length A on the y-axis, length N on the x-axis, and we plot all 3 combinations of the lengths there to see whether there's any correlation. So the heat map plots basically show the distribution. The darker colors represent more populated regions where the count is higher. So in all 3 cases here, we see quite a circular heat map where we've got a very dark center and then it sort of gets less hot as we spread out. And the sort of circular nature of them indicates that there's not much correlation between the 2 lengths. So if we were to take a fixed, say in this first example, a fixed length A, then this doesn't influence length N, for example. And then if we looked by residue, so we split up our histogram into the 20 different residues which we've got. we largely see what looks like to be roughly the same distribution for all 20 residues. There is an interesting plot for proline in the length A, and that is that it's just got this sort of slight peak at a slightly longer bond length there. But other than the proline plot, the rest of them look pretty similar. Then we can go on to look at angles. So again, plotting these histogram plots, and we can see relatively tight distributions. So we've got all the means there again, standard deviations, and we can see the percentages within 3 standard deviations of the mean and within plus or minus 1 degree. So again, the standard deviations are relatively low, but there's still plenty of examples where the bond angle falls significantly out of the sort of standard range. And if we sort of count actually in total out of our 704,000 chains in the cleaned PDB, if we count the number of examples where out of the lengths, at least one of the lengths or at least one of the angles is outside of 3 standard deviations of its mean, this corresponds to roughly 70% of examples within the within our 704K chains. So we can see that many examples basically have some sort of angle that's like non-standard. And again, we can look at these heat map plots and see very little correlation between the individual angles because they're quite circular and they've just got these dark centers as we had before. And then going on to look at the angle distributions by residue, we can see there's nothing too remarkable here really. For all of the angles, for all of the individual residues, the distribution looks roughly the same. It's just a single spiked peak at which spikes at the same mean in all cases. Where we have more variation now then is looking at the tau distributions. So it's worth noting here that for these histograms, we plotted slightly different ranges rather than the standard minus 180 to 180 degrees. And this was just because, well, for example, the tau CN plot here that goes from minus 90 to 270 now, if we plotted minus 180 to 180, then we'd be splitting the large 180 peak in half, basically, which, yeah, doesn't make it look as significant. So we've taken ranges that sort of don't intersect with the peaks. Yeah, so basically they go a lot across much wider ranges than the angle and length invariants, indicating that they're much more difficult to predict, and hence the structure of proteins is much more difficult to predict. Tau CN sort of shows these prominent peaks at 0, which is the cis case, and 180, the trans case. And one thing that I noted from the literature when sort of reading about these dihedral angles is it sort of suggested that proline has an increased prevalence of the cis case, so the 0 case relative to other angles. So by splitting into the various residues, I thought it would be good to investigate whether this is the case in our data. But first, before I go into that, we can also see the sort of, from these dihedral angles, the Ramachandran plot, which is the sort of conventionally plotted psi, which in our case is tau AC, against phi, tau NA. And so this is sort of, on the right here, we've got a plot which I got off the internet that sort of shows the energetically allowed regions for the backbone dihedral angles. and it's got plotted on it some of the sort of secondary structures that you might expect in each of the energetically allowed regions. On the image on the left is our heat map, so we can see it looks the same basically, which is good. We have sort of all three of these more common regions where we see more heat. Yeah, so it's quite good to know that, yeah, our results align well with this. But we can go even further than that, and we can plot all three combinations basically. So we'll consider the omega angle as well and plot all three combinations. So yeah, this one is just, we've changed the ranges slightly to reflect these new ranges that we're using, but that is the standard Ramachandran plot on the left. And then we've got the other two combinations basically. So we can see that from these plots that tau CN has sort of a less prominent peak at 0 and a more prominent peak at 180. So the cis and trans cases, as we mentioned previously. And in both of these plots, it seems that, well, tau NA and tau AC doesn't really depend on tau CN too much, because you can see they've got a range of values from across the entire sort of 0 to 360 or minus 135 to 225. Yeah, so there's not too much dependence there. If we split by residue, so I mentioned earlier that apparently sort of proline should have more, well, should be a bit different. So it sort of disagrees with the earlier literature claim really that proline is the main source of the cis dihedral angles for tau CN. So that is this plot on the right. And we can see that if we look at the distribution for proline, so this pink one here, that the two spikes in this one aren't really out of line in their sort of relative proportions relative to everything else. So we don't really see a peak of any particular interest there. We do, however, see sort of some geometric differences in tau NA for proline. So we see there's not so much of this peak that's around 45. It's basically non-existent here. And then this peak on the right as well is much more pronounced rather than some of the other distributions. We also see for glycine sort of slightly different distributions as well. Yeah, where the peaks are sort of slightly different, slightly broader, for example, compared to all the other ones, which look broadly the same. We can also do the Ramachandran plots from earlier by residue as well. So you can see these on the right. So the literature sort of suggests, if you look at Ramachandran plots, suggests that glycine should be the least restricted due to the fact it's only got one hydrogen atom on the side chain. And we can actually see this is the case from our Ramachandran plots as well, because we've got this whole region on the right-hand side that appears to have now opened up, whereas all the other plots mostly just have the sort of standard regions on the left-hand side. And then it also suggests that for proline, this should be the most restricted due to its 5-member ring side chain. So we can see that this is the case as well for this one. There's just sort of 2 very hot peaks, whereas virtually all the other plots have like a very hot peak sort of at the bottom, and then in the top left corner, they've got a much broader peak. So this is good. Again, it appears in this way to agree with the literature. So then moving on from the, well, sort of summarizing the distributions of the LAI invariants, we move to consider chameleon protein chains. So the word chameleon was basically referenced in this paper originally. So a database of chameleon sequences within protein science. But we offer a definition here. So chameleon chains, basically chains that are the same in terms of their primary structure. So they've got the same sequence, but they differ in their geometric structure. So in this section, we basically deal with identifying the most prominent example by defining a metric on the space of invariants, which will allow us to measure how geometrically similar two structures are. So this is what Olga previously mentioned in her talk. As the invariant for this bit, we'll use the backbone rigid invariant, BRI values, rather than the LAI values. And this is for the reason previously mentioned by Olga, where, you know, for the LAI, we've got angles, we've got lengths. So they're sort of not as compatible units. And it's much easier for this to use BRI because they're all in the same units. So we'll use this metric that was previously defined, the RMS BRI, where we can look at essentially how similar two BRI values are by looking at their BRI matrices. So for this analysis, I use just the most basic case, which is single chains. So what I meant by single chains in this context is chains that have no ligands in their corresponding PDB entry. They've got no sort of asymmetric units, and then their entries only contain one entity, and this entity only contains one chain. So essentially the most simple case. And the reason we did this was because we wanted to consider, if we want chameleon chains, we wanted them to be embedded in the same like environment, not be influenced by different ligands and things like that. Yeah, so we just restricted to these, which out of our set of 704K PDB entries, this was about 156K single chains belonging to 8.7K PDB entries. Of these 8,000 PDB entries, there are about 7,700 unique sequences, which we know is less than the number of PDB entries, meaning that some sequences were the same in different PDB entries. But yeah, we essentially restricted to just these 8,700 examples. And then in the subsequent slides, we'll take the examples of sequences which occur in the most models, PDB entries, and show their sort of geometric variability within different models in terms of the BRI. So we've got the 10 most prominent examples here where the counts are the highest. So there's just some primary sequences on the left-hand side here. Then we've got the PDB IDs that they occur in. So as we discussed, some of them occur across multiple PDB entries. So we've got this top one that's quite a prominent example that occurs across 16 different entries, but some of these only occur across one. And then we've got the number of models associated with all those different PDB entries. We've got the chain length, and then something that I'll discuss in a bit more detail is we've got the max RMS BRI between two of the examples. So this is, in the top case, consider all 1,700 models, and we look at all the comparisons of RMS BRI, and we can see, yeah, this is basically the maximum value. The two most different proteins. So we've got a number of plots here, really. So we're going to visualize the BRI differences between different models by plotting combinations of the BRI values. But in the case of this pairs plot here, because the BRI is this like large matrix of potentially m times 9 values, to distill it into just 9 plots, we take the average of each of the various invariants. So you can see plotted on the axes here, we've got the means of yn, the means of xn of those invariants. By taking the average, this means that if we've got 2 points that are the same on these plots, then their full BRI values might not be the same. But if their means are different on this plot, so they occupy different points, then they can't be the same, basically. So having matching averages is a sort of necessary condition of them being geometrically the same, but it's not sufficient. Yeah, so we've got this right-hand plot that, as I've said previously, sort of shows all the various combinations of the invariant means. And it's color-coded so that for the various PDB entries, we've got a different color in each case. So we can see from this plot here that some of the PDB entries sort of form their own clusters where they sort of seem to be geometrically distinct from other PDB entries, whereas some of the combinations of BRI values are altogether more random than that and just form like a giant point cloud, essentially. And we've also plotted here, so similar to the Ramachandran plot, we plotted for the LAI invariant here now, we can plot tau AC and tau NA, but this is just for the two most different examples. So in this case, it turns out to be this 2JZZ and 2KLG. But we can see from this plot basically that there's a wide variation in their tau NA and tau AC values. So it's color coded, so that one protein is in orange and the other one is in blue. And then lastly, I sort of went online on the PDB website to look at these two individual proteins that are the most distant, so the so-called chameleons. And you can just superimpose their structures on each other. So you can see that there's quite a bit of geometric, just visually, you can see there's quite a bit of geometric variation between the two structures. So I'm just going to go through the sort of 10 most prominent examples that we discussed earlier. So there's this other example, it's a very short protein. I think it's only 5 in terms of its length, chain length. We can see the plots here, so the various means for all the different examples. You can see the various torsion angles. So again, you know, you've got 3 blue torsion angles here that are vastly different than the 3 down here. And you can see the structures just don't coincide very well at all. Again, another example here where the structures just coincide quite poorly for the most distant ones. We see on this pairs plot all the different models that are there and the different sort of geometric configurations. So again, this one's quite nice for sort of separating almost into these clusters where for the 4 different PDB entries, all of the models can be quite similar to each other geometrically, but quite geometrically different to other PDB entries. Here's another example. So again, this is one that's spread over multiple different PDB entries. And we see, again, some of this clustering effect by color. We see, again, for the 2 most distant examples, that they are not very geometrically similar, and the torsion angles vary quite a bit. And then this is the last one that sort of occurs over multiple different PDB IDs, and it's the same story again, really. And we've got some of the 5 prominent examples that only occur within one PDB ID. So instead of the colors that we had previously for this pairs plot, it just varies by color by the different models now. So it's all just one PDB ID. But you still see quite a lot of geometric a lot of variation there within the geometry of the structure. And you can see the two structures are superimposed on each other once again, and the difference in the torsion angles. Yeah, here's just another one, very similar again. Another similar example. Fourth one, I think there's one more after this. Yeah. So again, many of these are roughly the same in that there's wide variation within the torsion angles and the actual geometric structure. So we can take all the examples. I just showed sort of 10 of the most prominent ones, but we can basically take all 7,700 unique sequences which form single chains and consider the maximal RMS BRI within each group. So that may be across multiple different models and multiple different PDB entries. And we can plot the results in the histogram. So we've got on the x-axis, we've got the RMS BRI between, or the maximal RMS BRI between two examples. And then we've got the corresponding count. So we can see that in the vast majority of cases that there are two examples that are quite geometrically different. So we've got this histogram peaking sort of around 1.2, 1.3. But, you know, there's a significant quantity of examples where the different models might have quite different structures, even within a fixed sequence. So, yeah, an overall summary of what I've gone through here. We've basically shown various continuous maps based on our cleaned version of the PDB. Whilst many of the geometric parameters, such as bond length and bond angle, are generally thought to be well determined, there are plenty of outliers, as we've shown, with non-standard values. We can see from the Ramachandran plots that show 2 of the 9 LAI values. They're basically insufficient to determine the geometry of a protein, and we need 7 more variable parameters to consider. The identification of these chameleon protein chains show that even knowing the primary structure of a chain may be insufficient to predict its geometry, and different models of the same protein can be quite geometrically different. And we should sort of aim to move towards a world really where we're not afraid to discuss all 9 LAI values or BRI values when we consider the geometry, or when we compare the geometry of 2 protein chains, as these are basically required to determine the geometric similarity up to rigid motion. Yeah, so that's it. So any questions?
Yeah, thank you.
Oh, sorry, let me turn my microphone off.
Thank you, Will. Yes, thank you for thanking Will. Let me stop the recording.